it? We have to start? Good morning. Good morning, good morning, good morning. And we're going to say toodles to the Doug Cam because I have learned that I cannot conduct polls if I have the Doug Cam going. Ah. All right. Well, welcome. Today is the last day for mollusks in the lecture. It's a sad face for me because they're kind of amazing. But maybe it's a happy face for you because you think, well, I've seen pretty much all the mollusks can do. And I'd have to disagree. There's more, and we're going to talk about that particularly. So today we're talking about bivalves. We're going to spend most of our time with bivalves today. And then scapepods, which is the other of the 8 principal classes of the mollusks that we have visited. And I will keep my eye on the chat here and on the door to keep letting people in as they arrive. So, and also happy St. Patrick's Day to anyone amongst you who might be celebrating such a thing. All right, so that's it. Today is the bivalves and the scapepods. Now, the bivalves are a diverse group. Particularly, we live in kind of one area where they're actually particularly very diverse. We, meaning us, and this area, meaning kind of where I am. And not where all of you are, but where some of you are, in the Grand River watershed specifically, and in kind of eastern North America in particular. Now, the synapomorphies for the bivalves is that we've seen them before. I actually laid out a picture to compare and contrast them with the brachiopods that we saw back when we were dealing with the Lophophorata. And we talked about the difference between shells that are laid down like this versus laid down like this, so dorsal-ventrally versus laterally. That's the principal difference. And millions and millions of years of evolutionary history leading to that similarity. So they have a bivalve shell with a hinge up there. They have adductor muscles for closing the shell. They have no radula. So this is another case where one of the principal defining synapomorphies for the group in general is not seen. Leave it. Leave it. Please don't pick apart your duck, because then I'll have to buy another one. The principal synapomorphies for the group is lost in a particular interior group. So no rasping, that kind of ancestral view that we saw like a monoplacophoran or a gastropod, that is not here. Most are suspension feeders, and they have adaptations of the gills associated with that. They have siphons. The mantle edge is very plastic here. It does a bunch of things. We'll talk about kind of 3 specific things. And they're relatively long-lived, and you can compare and contrast that with the cephalopods that we saw last time. These ones are not nearly as cephalized, not nearly perhaps as intelligent, but can live for 150 years versus kind of a year or less, which we see with the much more intelligent cephalopods that we spent time with on Monday. So the gill lamella. So looking at different kind of aspects from figure 13.8 of your text, different morphological aspects of this group, and you've seen some of them already in your lab, I realized last week, but we've got this dorsal umbo bent over the hinge, and then peeling open one of those valves, we can see a bunch of the different apomorphies for the group evident in that internal visceral mass. So first of all, we've got the mantle, doing a bunch of different things. One of them being that it's secreting the shell, protecting. It can be sensory also. We have 3 different folds. We talked about that one time already, and we'll talk about it a little bit more today because there's some spectacular adaptations that bivalves have within their sensory, their middle paleo fold. So they have an inhalant siphon, and these gill lamella, and then an exhalant siphon. Exhalant. It is most excellent, but it is also an exhalant. Then the foot a large ventral foot. And so this is each of the valves, each of the two valves has a dorsal peak called an umbo that rises up over top of the hinge, and that represents the oldest part of the shell. So it's being laid down kind of distally around that. Smith turns back to the door. Welcome, welcome. Today we talk about bivalves. And there's the dorsal umbo there. So the mantle cavity, so we've switched perspectives here. So now we're looking at these left and right hinges and the hinge ligament. And you can see in this section, the foot hanging down like a long kind of dangly tongue in the middle of that mantle cavity. And then we have these branched lamella. And we're going to talk a lot about, there's different kind of complexities that they can have to these almost like hanging, blinds or drapes that are hanging, suspended within the mantle cavity. Because you can imagine the mantle cavity, the capacity to bring in water remains important for the gills in terms of providing oxygen to the blood, but in this case it's doubly important for the bivalves, and doubly important, and you can imagine turbidity, increased turbidity in the water being doubly damaging when you are also, not only are you trying to respire out of that water obligatorily, but you're also trying to feed out of it. So we've got that large mantle cavity and then the mantle skirt hanging around the outside. Now, right at the tip of that mantle skirt, we have the paleofolds, and this is kind of the edges of the mantle that you've seen before. We talked about this figure, 13.15 before, because we talked about, show my three fingers to show the muscular mantle lobe, the sensory middle mantle lobe, and the outer secretor. So that's what's secreting the shell, the peristracum. We've got also in this the three layers of shell, the nacreous, the prismatic, and the periostracum, so kind of from proteinaceous to prismatic. We saw prismatic, remember, with limpets with photonic colloidal cells embedded there, and then a nacreous and underlying nacreous pearl-like layer. Now, the periostracal groove, so there's the three of them. The periostracal groove is kind of a cool thing right in here, this extrapaleal space. Some of you probably have the periostracum seals off that extrapaleal space and isolates it, and that's in fact where, if there's tiny little bits of sand or other kind of material that gets itself in there, the shell, that mantle layer that is secreting the shell will actually try and secrete material around that irritant, and that nacreous layer that it lays down, left years after years after years, can become these pearls. So there's that extrapaleal space. Now, I'm going to talk about the middle fold for a little bit and talk about some recent, some things that we've known for a while, and a recent study that has kind of blown up what we thought we knew about it. Now, one of the cool things about the extrapaleal fold is that it's a sensory, as we talked about it, and has a lot of eyes. I'm going to go back to that last slide. I apologize, I put these two out of order. But this is looking down now at the fact that there are light sensing eyes, up to 200 of them, around the edge of this scallop shell, this bivalve shell, this mantle. And so each of these individual eyes, 200 of them, we've known about them for a while, for upwards more than 100 years, but they're very small. These are less than kind of a millimeter in size, very, very small, tiny eyes. And we thought that they're principally just about sensing light. So firing those adductor muscles, closing the shell, protecting the vulnerable, the foot, but also the very vulnerable and important gill structure, branched gill structure. We've learned some more about them. Now, in this video that's looping through on the bottom here, associated with the science paper from a couple of years ago, there's a diagram here after we see these gorgeous blueberries. There's 2 retinas within this eye, and then a prismatic mirror layer. And that's one of the things that differentiates these individual cells, is that up to 200 individual eyes, that as light enters the eye, it's very, very different than either of the other mollusc that we've talked about together. Remember the polyplacophorans, the chitons eye created out of shell, or the cephalopods eye created and analogous to our own. This has a pupil, a lens, 2 retinas, and that's what you saw in orange and blue in the last slide, and then a mirror. And the mirror is made of crystals of guanine, and that's these kind of regular eyes. Instead of being prismatic, they're flattened crystals of guanine. So that's kind of amazing already. So we've seen an eye made out of analogous to ours in the cephalopod, that looks like ours, that acts like ours, camera-forming eye. We've seen other eyes formed out of minerals, essentially rock eyes in the polyplacophoran. In this case, not to bend too far a point, but you know guanine, it's one of the 4 principal bases that make up DNA. So we're seeing a DNA mirror-based eye and that inner mirror is curved, and then neural signals are generated and sent to a small visceral ganglia. But this structure looks not at all like ours in cross-section. It looks much more like what modern segmented mirror telescopes look like. In terms of photoreceptive capacity, they have 12 opsons, we have 9. So even though they probably are seeing things within blue-green because they're underwater, they potentially are seeing colors that we cannot see. And one of the neat things about this most recent study is that it demonstrated that pupils are able to open and contract. And so you can see an open on the right-hand side, closing, and a much more closed pupil as light intensity went up. But if you can kind of tilt your head, you see that's kind of 15, 0.2, so 15 seconds, and then a minute, and then 10 minutes. So it's not fast. But it is able to contract. So these 2 retinas, we think, are not just they're more complicated than we had historically thought, just being shadows. These 200 eyes with their DNA mirrors likely produce 2 different images. So they're seeing from each of the 200 eyes 2 different images. One of them, the larger one, the orange here and the ring one, it's likely about the periphery, seeing from edges. And then the smaller one is about clarity, so potentially actually seeing things. So scallop eyes in mollusks, that's amazing. It's amazing. I hope you share that excitement. So a little bit more about the shell, that we've got these valves that are articulating along the dorsal hinge. We've got hinge ligaments, and then 2 adductor muscles that are fast twitch. And when the valves are closed by the adductor muscle, which you can see here, the outer hinge ligament is stretched and the inner ligament is compressed, and we see that the shell is open. And then and so you can see that it's firing across those ligaments that are essentially acting as hinges and helping it spring closed and shut. And so having 2 offset springs, helping it respond very quickly to potentially nibbly feeding predators. Now the foot, that large tongue hanging in that mantle cavity, it can be used to anchor and to move around. Some bivalves, including scallops like we just saw with their 200 eyes, they can actually use those adductor muscles to kind of bellow the valves open and shut and swim. And perhaps you've seen video of that. But the circular pedal muscles contract, and that, just as we've seen with a lot of our different hydrostatic pre-mollusk, practically all of the other organisms with a hydrostatic skeleton. We talked about antagonistic muscle layers, and so it extends as the circular muscles around that foot causes it to, in the portion A, to extend, and then different firing anchors it, and that can then pull the mollusk, the bivalve, under the surface. So just the inhalant and exhalant siphons are above the water, thereby, as you can imagine, providing a lot of different protection. And there's the description of that you can read later on. Now that foot is, we talk about as a pedal sinus, but it's essentially there is a, now wait a second, there is a small kind of coelomic extension in there. The other thing that the foot is doing is laying down byssal threads. So what you're looking at is kind of the foot extending out of this shell, and what you're going to see in this video is it start to throw these byssal threads. Those of you who live, and there's a large plaque connecting it. My apologies with my jumping on the mouse. So, as it's laying down these byssal threads, if you've run into or tried to pry mussels or other bivalves off of rocks, if you've lived coastally or either freshwater or marine coastally, you know that they can attach themselves very, very efficiently. And that's accomplished by these byssal threads. So we see the byssal threads extending out produced by this gland. So each byssus is a bundle of threads that are tipped with little adhesive plaques. And then there's tension, so they can change the angle that they're sitting at according to where the force is coming from, potentially with different tide levels you can imagine in the ocean. And these different byssal threads and their adhesive plaques are then laid down and sent out and attached right on to onto the rock. And so the location of that thread in the plaque is biased towards the plaque's proximal edge. And so physicists and engineers are really interested in this from a couple of different perspectives. One of them is the adhesion capacity for the plaque, and the other is in terms of how they lay down the byssal threads to deal with different forces from the water. So you can see they're laid out quite flat if the water is immobile. If there's a lot of lift, like based on where they're sitting, if there's a returning wave action, they spread them out. And then if there's a particular directionality, they accommodate for that in laying out the threads into the force, into the current, which is kind of amazing. Remember, this is an animal that is not cephalized at all. These are responses, kind of responses to an environment. I mentioned that engineers having a lot of interest in bivalves and byssal threads, and a lot of that has to do with bioproducts. And this was a paper from a couple of years ago in Proceedings of the National Academy of Science, PNAS, where they looked at ferric-mediated bridging between protein films in nature inspired by mussels. So a self-healing process that involves metal ions and proteins that are all about how that mussel is keeping it attached to otherwise very either slippery in this case or not pocked or creviced at all, like this bit of aquarium glass that you're seeing in the video. So again, my suggestion to you, if you're at all interested, if you have an engineering bent and you are being inspired by, I hope you're being inspired by all of these kind of biomimetic solutions that millions of years of evolution have come up with, that we might be able to use ourselves for inspiration. And thank you for asking again. I am recording. It is, and yeah, that was a big help last time when you asked whether I was recording. And I don't know how many of you have gone back or are attending asynchronously and you just saw it, but I hope that seam in between. So I had to re-record the first part of the lecture and then stitch them together and make a Franken lecture. So thank you for noticing that. And I'm just staring at it now yes, I see it ticking over. We are recording. Now, about the gills and feeding. So there are three different, we'll talk about, as we have with many groups, the increasing in the size, dealing with the increased surface area, increased size of the gills, resulting in increased surface area in this case, allowing the construction of a larger volume of mollusk because it produces a more efficient amount of area that can both respire and trap particulate matter, so it can eat. So within, we have protobranchs, lamellibranchs, and septibranchs. And these all have to do, if you take a look at the number of kind of folds that are created. So if we start with the protobranch where we have just kind of two mantle nidia that are hanging off on each side. And then we have a lamellibranch where there's an interlamellar junction that produces two different kind of folds. And then we have a septibranch, and they follow a very different, they eat very differently, and we'll talk about that on its own in a second. But, so a word about these intralamellar junctions. This is this tissue here that's bridging between the ascending and descending arms of the same filament of the gill. So it's an interlamellar junction. And that's allowing there to be more folds, more surface area. And you can see we talked about that inhalant and exhalant canal, and I'll show them to you here. So we've got ciliary currents. This is a trait that we've seen with a lot, or this is an ancestral trait of the mollusks that is taken to a large degree here, taken advantage of to a large degree here in the bivalves. So the direction of the ciliary currents in the mantle cavity and on the palps. The water currents we also see here because they kind of arrive and depart, inhalant canals. And so as the water is brought in, it's brushed across the mantle gills. There's the exchange of oxygen, but there's also in these ciliary currents the capacity for extracting small bits of food from the water. Now septibranchs, with this large hanging, so we've got the foot and then a very reduced gill. This is because these are essentially raptoral, they're carnivorous of a big scale. So we see that they have an inhalant and exhalant siphon, but this real change in morphology associated with eating large prey items. And they do that using suction that's generated by the mantle cavity. So as we go back to proto and lamellibranchs, these kind of the classic bivalve feeders, we see, if we look along the gill folds, right here, you can see there's a food groove labeled in that SEM, the micrograph at the top. Now, we have then the mucus string. We've seen this with a whole bunch of different mollusks. So we've got currents coming in and ciliary action, kind of folding food down into that food groove that then feeds it towards what looks like a cauliflower in the center and the mouth. So the lamina of the labial palps then kind of feeding the food right into the mouth. And then we see the same kind of classic, this is actually a bivalve that you saw earlier when we looked at the classic digestive system of this mollusk. So we've got a ciliary sorting fields with a chitinous cuticle, ciliary sorting area, and the typhoseals, and then a crystalline style that's drawing mucus and pulling it through from, I did it again, I've showed you from the, but I'm kind of showing you with the wrong hand, but drawing it from the mouth and sending it out towards, at the end, towards the anus. So high surface area but low efficiency. This is the gas exchange for the story for bivalves. The mantle skirt can also, they've increased not just where they breathe, restricting it to the gills, which are kind of doing, as we've said, two jobs here, but there are different folds in the mantle skirt that can actually respire. So not just can the mantle create the shell or allow it to see, but it also helps it breathe. Perhaps there's nothing that the mantle cannot do. The respiratory pigments that we've seen, some have hemoglobin, some have hemicyanin, and Remember, we've talked about these respiratory pigments before. We've got the iron-containing oxygen transport metalloprotein hemoglobin. Hemicyanin is the one that's greenish with copper, and then hemierythrin, which has a pink or a purple color. And their distribution we've also talked about phylogenetically. So within the mollusks, we see principally two of them, the hemicyanin and the hemoglobin, and then hemierysthrins. And so thinking phylogenetically, and I've already in that last lecture quiz, I've asked you to think kind of phylogenetically about it. How do you explain frequent origins, one origin, multiple losses, multiple origins, and then how do you explain function in lineages that have without circulatory systems? There's a whole bunch that we still have yet to properly understand about the distribution of the evolution and distribution of respiratory pigments. So the mantle skirt. So internally we have the intestine. Sorry. So we have there's, as I said, respiration with the mantle skirt. And then just finishing up the description of that digestive system, the intestine through the pericardium ventricle, and then the blood essentially functioning as a hydrostat. Remember, we've compared and contrasted several times different groups of mollusks with an open or a closed. And the closed, we kind of asked you to align your thinking with the cephalopods, thinking about mobility, and so thinking about not just having a large sinusoidal cavity where organs are bathed in blood and respiration is relatively inefficient, as it is here in the bivalves, but with the cephalopods associated with greater mobility and their pelagic lifestyle, that we saw the evolution of a closed circulatory system that allowed that efficiency to happen, that one couldn't have happened without the other. Their nervous system.
The scallops are also
there we go.
They live on the surface and not only have good eyes,
there's
but
the
a quite
small eyes.
surprising way of getting around.
And here's the view of the swimming. So our bivalves swimming through the water. So less cephalized, then they tend to be more sessile. The sense organs tend to be concentrated in paleofolds, and in fact, the paleofold of the mantle can even be extended into tentacles. That, as you can see in the bottom here, these are extensions of the mantle that have ciliary receptor cells that can be mechanoreceptors or photoreceptors, chemoreceptors. We have, as we've talked about, paleo eyes, cerebral eyes. The other part, the other synapomorph for mollusks that they retain and is quite important to them is this osphradium, which is in the excellent chamber of the mantle cavity that deals with chemoreception and mechanoreception, and showing it, telling it kind of when to fire those adductor muscles and close, perhaps not based on predation, but just on the turbidity and the potential damaging capacity of water that's full of particulate matter. Now, the mantle is not done. Some of you may have come across this particular animal before, the disco clam. And you can see that fold of the mantle. You can see why perhaps they called it the disco clam. So this is an organism that doesn't have well-developed eyes. This is a particular bivalve that doesn't have well-developed eyes. But it has, one of the folds of the mantle is apparently producing this rhythmic flashing. And what I would like to do for a second is to ask you to jump into the chat and tell me some ideas about why you might see a relatively sessile organism with not a highly developed set of photoreceptors. What role is flashing playing there? Pop those ideas, if you would, please, in the chat. And as you're doing that, I see that there's some, is it able to respire on land using the mantle? Not well, like it wouldn't live long, but it can respire for a small amount of time. When will we be getting the grades for the second lecture quiz back? As soon as everybody has completed the quiz. There's still some people who have not. All right, and now we have some ideas. Okay, to attract prey, signaling to others, mating, defense against predators or to ward off predators. So tell me about, so I think there's kind of three, I think there's three things there that I'm seeing. So tell me about how it might be useful for mating. Tell me about how specifically you think it might be useful for prey attraction, and what might the flashing do to defend against predators? Have you got, expand your ideas. I like them. Get out. Silence. Doug. Okay. As you're expanding your ideas, what I'm going to do is I'm going to try and project a poll here for a second. I am not logged in on another device, but... Or am I? Okay, let's try that again. All right, so the poll was going to ask for a show of hands, basically, for of these three principal ideas, mating. Let's try and conduct this in the That's a great idea, Sherry. Look at that. We have such like multifaceted communication. I do. We may see the poll here in a second. It's the very last poll. 20th or 20-something. I think it's called something cryptic like disco clam. Okay, so let's deal with some of your thoughts here. So signaling to others, attraction of prey, and defense against predators. So the flashing could confuse the predator. I like that. So signaling to others could be alerting those around them of predators that are around. Perhaps the flashing makes predators think it's toxic. That's interesting. If it has no eyes, it might be able to use the flashing lights to attract prey, similar to deep sea fish. When the prey get close, it just then uses maybe those paleo. That's a great hypothesis if it's carnivorous, and broadly speaking it is. Not in a big septibranchial way. For mating, it could be that there is a best fit for the environment that attracts either others or attracts the fish they need to implant the eggs. Ah, you're retaining some of that, the stories about bivalves from 2700. This is 2700, from 1070. That's interesting. For mating, the light could actually give away the clam's location, which means it makes it more vulnerable. So the clam then has to survive while drawing the kind of attention to itself. It shows it has stronger genes. Stronger genes. I like these hypotheses a lot. I'm guessing that we haven't had success showing the poll.
Alex, the poll is showing, and I'm guessing you can't see it. I know I can't see it when I'm running them either. But So we've got 85% of the people voting.
That's awesome.
And what role does flashing of the disco clam play? We've got 17% said sex, 43% said food, and 38% said warning.
I love that. Now, can you display it so it appears on the screen and I can get a screen grab and insert it in the PowerPoint later on?
Like share my screen?
No, share. But I think with the poll, there's a command like share poll results.
Oh, let me end the polling and then see. Yep.
Thank you very much, Sherry. This is awesome.
There, can you see it?
That's so cool. I'm guessing that people can. Tell me in the chat if people can see it. I can't actually see it. So weird. All right. I will draw something. Thank you very much. Hooray. So 43% of you thought, which was the 43%? The largest was
Food.
Food. Okay. And then 38% thought predators, and the 17-ish% thought, that's really interesting. So let's kind of unpack this a little bit. So the marginal edge of the fold, because we do know a little bit, as of kind of 2 years ago, we know a little bit about this story. You're looking at a really slowed down, 1,000 frames per second here, is that this signal is essentially being sent out, and we see the flashing light that appears there and there and there on the marginal edge, and then it furls up in kind of a wave-like action. And what's happening there is that there's highly reflective silica spheres that are in that mantle edge. And in fact, I'm going to come back to that. I'll come back to this, but essentially, the hypothesis is that this is kind of like an aposematic thing, that the silica spheres themselves don't do anything to a potential predator. But the current hypothesis is that we're seeing that the retention of this disco clam, this disco-like silica extension, because they're sending a somewhat honest signature of unpalatability to potential predators. So it's not about communication intra-specifically to for sex. It's not so much about attracting potential prey items, but it's about telling predators that it's like a red warning signal, or like the coral snake. It's a signal that I'm an unpalatable thing. Or like the monarch, excuse me. So like the bright orange of the monarch. And in fact, it's an honest signal. It's not just taking advantage. It's not just kind of flashing the lights and it has no bite to that signal, because there is an acidic mucus that is expelled when they're handled. And there's actually so an aposematic, so an honest warning signal. And this paper that came out a couple of years ago demonstrates the fact that there are, this wasn't the first one to demonstrate the reflective, the silica spheres, but this was the first one that kind of pulled apart the fact that there are compounds that are highly repulsing towards different predators, like the mantis shrimp. So the disco clam, amazing. Now I'm actually looking at the chat right now, and I see that there's, after your last flurry of fantastic suggestions, I don't see anything newer, so thank you for that. So reproduction and development, this is something that you're actually familiar with, because everybody who's kind of emerged through 1070, and you can all say hello to Dr. Jacobs as they walk by in the background, where we talk to you, either the two of us together or just Dr. Jacobs talk to you about mussels in the first unit of 1070. And we talked about the fact that there's some amazing adaptations that different lineages have. So bivalves are gonochoric. There are males and females. They're externally fertilized. They have, like all or most of the other, they have the characteristic for the mollusks, the synapomorphic, the trochophore larva with a little mustache. They often have a villager larva. And some of them have another different larval stage called the glaucidium. And the glaucidium is the one that we talked about at length within 1070, talking about their attachment to different fish species. And so there's some external fertilization of a classic kind of a mollusk happening. The release of reproductive cells into the environment.
In the streams of Missouri lives the Lampsilus mussel.
And this is a video about that you can go and watch later on. I'm going to mute it for a second, kind of jump around. But these are the Lampsilus mussels that we talked about in 1070, where they are using this, a couple of different things. Again, with an adaptation of the mantle. Remember, the mantle fold here has become essentially a lure. And that lure brings in particular species of fish, species of mussel, luring particular species of fish. They then slam shut on the fish head and expel larvae into the gills. Those larvae, the glaucidium larvae, that little snap jaw that you saw earlier, they grab on to filaments of the fish gills, and then essentially exist as blood-sucking parasites until they drop off and begin to eat on their own. So this is imitating prey species. So it's kind of a tritrophic interaction where the mussel is attracting a particular species of fish and then bam, releasing those Glaucidia larvae. So this is one species of mussel imitating another species of fish that the desired host species is then attracted to. And then you see the little Glaucidia larvae grabbing on to folds, nice blood-rich folds of the fish gill. Yeah, wow. I love talking about that in both of these classes because that's bananas. And that's a lot of the diversity that's here, or that was here. And I hope we'll be able to finish off by talking a bunch about the diversity. We've seen a diversity of different styles of life within that. We've seen freshwater bivalves. They can exist as commensal or parasites. They can be boring, where they drill in. They can act as attached epifauna, obligatorily attached to the surface, or unattached, where they can move around a little bit more. So we've got attached and unattached. So imagine for attached picture, maybe the super encrusted zebra mussel, and then the unattached, think about the scallop. So it's kind of moving itself around either with the foot or with the jet action of the bivalve shell. Now the boring mollusks, so shipworms. This is another use of the word worm to indicate this long vermiform-like extension. So we've got the foot, the hood, and then a long fused mantle fold. So again, the mantle doing another thing here that's essentially acting where the valves of the shell are being used as a drill. And they used to be incredibly damaging, economically super important because of the damage that they caused to shipping industries, because of the damage that they would cause to when ships were wood, to the ship themselves. But then also, and for a longer period of time, as we transition away from ship more to metal hulls, docks remained wood for the longest period of time. And they could be, you can see kind of the result here in a Texas bay and a Texas harbor of a shipworm infestation that caused essentially the collapse, not just of the dock structure, but of the adjacent houses. And so in freshwater, we've seen a bunch of different adaptations. There are some, like the zebra mussel, that maintain the planktonic veliger larvae, so they can disperse over long distances, and they were kind of an ideal strategy or an ideal organism to, unfortunately, travel to our continent in ballast water of ships from the Black Sea in different parts of Europe and infest our watersheds, towards the lures and the highly species-specific other taxa that we talked about. Functionally, bivalves play an enormous role in our freshwater ecosystems. And in the last kind of couple of minutes, before we get to kind of a quick tip of the hat to the scaphopods, this is what I want to talk about. This is an older paper now, but it basically categorizes a lot of the functional role, the highly important function that bivalves play in freshwater ecosystems, where they filter phytoplankton, bacteria, and other particulate. That has an enormous effect on nutrient dynamics, both through the filtering, but also through their excretion, through the biodeposition of feces and pseudofeces. Pseudofeces is essentially non-digested material that's wrapped up with mucus that gets expelled out of the anus of the bivalve anyhow. So when there's bioturbation of sediments, the bivalves can increase sediment, water, and oxygen content, and then release nutrients from the sediment into the water column. They can free things out. They can also act as repositories of those sediments. So a lot of the toxic chemicals that might exist within Lake Erie, the highly efficient zebra mussel filtration system has actually capped it and contained it within a bed of zebra mussels, which hasn't removed it from the system, but it's essentially strated it and made a layer on the deep water and on the substrate. The physical presence of shells, speaking about zebra mussels, can create habitat themselves. And then the arrival of Dreissenia, of the zebra mussel, has created associated with pollution, and anthropogenically altered freshwater ecosystems in eastern North America has resulted in a catastrophic decline in benthic biomass and diversity. And so what you're looking at here in this little movie is kind of through time, based on sampling data, and you can see the stripes associated with major riverways. But from 1981, the spread of the zebra mussel by human-mediated forms, by essentially us with bait water, with uncleaned water within boats, traveling between watersheds, having not likely adult mussels, but larvae, villager larvae that can then expand and colonize that environment quite drastically. And so there's a large, if you're interested in conservation biology and you're interested in this kind of Eastern North America part of the world, a lot of the endemic diversity that we have are our freshwater mussels. So like the wavy-rayed lamp mussel, Lampsilus, this is one of the lure-bearing ones that we talked about earlier. You can read up about them on the committee on CSIWIC, so the Committee on the Status of Endangered Wildlife in Canada. These are some of the few species of invertebrates for which we have CSIWIC assessments and status reports written. One last nod in our own watershed, a study that was published a couple of years ago. So we're sitting, or I'm sitting here in Guelph near the intersection of the Speed and the Aramosa rivers. Once the Aramosa dumps into the Speed, it flows downstream into the Grand. The Grand is then the principal watershed on this north shore of Lake Erie, and it flows down near Dunnville and enters into Lake Erie there. And what these researchers discovered was essentially by sampling along the length of the Grand, which essentially starts out as principally agricultural, and then as it flows through the Kitchener, Waterloo, Guelph area, becomes increasingly urbanized, that mussel abundance changed dramatically, mussel abundance above the city was quite dramatically higher than it was downstream of the city, that mussels downstream of the city were dominated by larger size classes, suggesting a zombification, that it's the older ones that are living and that younger ones are not replacing. So we're kind of on the edge of likely the further catastrophic loss of species and biomass. And this has a lot to do with wastewater treatment and homogenization of shorelines. I can see the chat from where I'm sitting, so we're just going to keep going to spend the last couple of minutes talking about scaphopods. This is the last of our mussel groups, the tusk shells. So scapho is Greek for spade. So you can see where the scaphopod name came from. So they're tusk-shaped conical shells that are opened at both ends, and they're characterized, one of the synapomorphies for the group is this captacula, which are these anterior thread-like adhesive feeding tentacles. You can see them here. So these are the captacula. So their primary responsibility is feeding. And there is a small video. The YouTube and Vimeo are not full of a lot of good scaphopod videos. Maybe you can go out and change that. But this is video of the anterior end of a scaphopod extending and moving its captacula. So conical the shell is and slightly curved to the dorsal side, but we've got the foot anchoring it, the captacula extending out, and then a shell opening with water entering here, excuse me, being brought down across the mantle cavity. A reduced gill, so there's respiration happening on the mantle here. So again, I guess one of the themes for, pardon my coughing in your ear, one of the themes in general for mollusks, and maybe in particular brought into tight focus in this bivalve and scaphopod lecture, is the practically near plastic complete capacity. If a mantle is given enough time, it can practically do anything. It can see, it can move, it can secrete a shell, and it can respire. Excuse me. And so that is it for the mollusks, and that is it for the Spiralia. So this large group, the outstanding small phyla will kind of catch, I'll give you basically a one slide description of them later on in the course, but that's the dedicated time that we're going to spend with the Spiralia. For the next big chunk of time, we're going to jump into the ecdysozoans. This is the most diverse group. It includes nematodes, crustaceans, arthropods, tardigrades, and onychophorans. All of those things could be courses on their own. One of them is in terms of the insect biology. So that's it. Thanks for playing along today and sharing our enthusiasm for mollusks. And I'm going to, as I transfer things to the interpipes, I'm going to stay in the chat. So if I did miss a question, please re-up it, and I will deposit kind of the page's worth of links there, and you can go and check them later on. So take care of yourselves. Enjoy St. Patrick's Day. Don't drink green beer. That's ridiculous. If you're going to drink beer for St. Patrick's Day, have a Guinness or a Jameson. Have something sensible, a sensible liquid from Ireland. And we'll see you Friday.